A major advancement for reaction center research was made when the R. viridis reaction center was characterized. So in 1984, a group of scientists used X-ray crystallography to analyze a membrane protein, and this membrane protein happened to be from a photosynthetic purple bacterium known as R. viridis. In particular, that membrane protein was the reaction center R. viridis. Now we're going to take a look at its structure. The first subunit is ethyl, shown in blue. It has 273 residues, 5 membrane-spanning alpha helices, 2 molecules of chlorophyll, and 1 molecule of pheophytin. Next is subunit M, shown in yellow. It's basically the same as L, but it's a little bit bigger. It has 323 residues. Next is H, shown in orange. It has 258 residues, 1 membrane-spanning alpha helix, and most of the protein actually forms a globular domain in the cytoplasm. Next is cytochrome. It has 333 residues, 4 heme groups, an N-terminal cysteine residue, and is linked to the membrane by 2 fatty acid chains. This figure shows all 4 subunits together. One important thing to note is P870, which is made of 1 chlorophyll molecule from the M subunit and the L subunit. It's responsible for absorbing the light. Photoexcitation of P870 leads to an electron being transferred to BCHL and the production of P870+. BCHL then transfers an electron to QA, which then transfers an electron to QB. QB flows to cytochrome BC1, where it is reduced. Reduction of QB is coupled with the translocation of protons, which helps to establish a proton gradient. Cytochrome then transfers an electron to P870+ to restore it to P870. Now we'll look at the molecular architecture of PS2 in a cyanobacterium. PS2 is important because it accomplishes the light-driven formation of oxygen from water. This is the structure of PS2 and T. elongatus. Some of the important things to note are P680 in the middle, which is responsible for light harvesting, the 2 tyrosine residues designated Y, the OEC, and the iron atom coordinating the quinones. This slide is going to compare the structure of PS2 and T. elongatus to the R. viridis reaction center. One of the important similarities is the pattern of electron flow through the reaction center, which I'll talk about in just a second. This slide is nearly identical to the one that we looked at earlier. Some of the main differences are that before diffusing to cytochrome BC1 to be reduced, QB receives 2 electrons from water and 2 hydrogen ions from the stroma. Also, the OEC transfers an electron to P680+, which is coordinated by one of the tyrosine residues. This is the structure of the OEC. The active site contains 4 manganese atoms, 1 calcium atom, and 5 oxygen atoms. This cluster is stabilized by various amino acids shown in the figure. As protons are liberated from water upon its oxidation, they diffuse into the lumen where they contribute to the proton gradient. Next, we'll look at the molecular architecture of PS1 in cyanobacteria. This is the structure of PS1 and T. elongatus. Some of the important things to note are P700 in the middle, which is responsible for harvesting light, A0, which is another chlorophyll molecule, A1, which is a phylloquinone, and all of the iron-sulfur clusters. This slide compares the structure of PS1 and T. elongatus to the R. viridis reaction center. Again, one of the important similarities to note is that there is a similar pattern of electron flow through the reaction center, which I'll talk about in just a second. This slide is very similar to the other 2 we looked at earlier. Some of the main differences are that electrons flow through 2 of the iron-sulfur clusters to ferredoxin. Ferredoxin then diffuses to ferredoxin NADP+ reductase. Also, plastocyanin supplies an electron to P700+. Now we'll examine the molecular architecture of both photosystems in higher plants. So now we're wondering, do higher plants follow the same architectural patterns we've been talking about? And the answer is yes. Based on new structural information about higher plant photosystems, the same architectural patterns are followed, and we'll examine the architectural structure of one of these photosystems in just a second. This is the structure of the PS1 LHC1 supercomplex. It's basically like a really big version of the PS1 found in T. elongatus. It has 173 chlorophylls, 2 phylloquinones, and 3 iron-sulfur clusters, as well as a lot of alpha helices.
Created using PowToon.